Aloha, my name is Geraldine Milla, and I am a rising junior at Kapiolani Community College under the NSAM PEACE 2 and MTC program, majoring in computer engineering with my partner Nanea.
Hi, my name is Nene Okiolasiu. I am an incoming freshman under NSEM's Freshman Bridge program, majoring in biochemistry.
Today, Nanea and I will be presenting our research on image analysis using deep learning under the Micro Devices and Microfluidics Laboratory advised by Dr. Aaron Ota and mentored by Meenakshi Vora.
A little overview about what we'll be touching on today during our presentation is our research background, expectations, a little project overview about the steps we've been taking to complete our project, and conclusion. So, in the beginning, we trained with analyzing the size of embryos from videos to familiarize ourselves with how to complete analysis and image labeling for the droplet project, which helped us gain an understanding on how to solve the droplet problem of identifying a droplet when given a video so that our computers are able to identify it without supervision from us. And this set the stage to work on our droplet project. So as you can see here on the left is a image labeled microembryo, which we went and image labeled the difference between the embryo and the background so the computer can be able to differentiate for itself. And then on the right, you can see we successfully had a trained microembryo that the computer was able to analyze by itself. Why we're doing this project, the benefits of this project is to be able to identify cancer cells sooner. In Hawaii, over 6,800 residents are diagnosed a year, and over 2,000 of them die from cancer. So we want to be able to catch these disease cells at an earlier stage. And overall, this will help cancer treatments cost because right now it costs up to $678 million annually to treat for cancer. So we want to be able to lower the cost and lower the diagnostics rate. Skills acquired during our research were being able to analyze a research problem and getting to the solution. We also use programming skills and tools during our research, which were MATLAB, which we use to run our codes through, and use image labeling, image analysis, machine learning, and deep learning, data acquisition, and data analysis. And what machine learning is it focuses on the development of computer programs that can access data and use it to learn for themselves. It's an application of AI, essentially, artificial intelligence, that provides the systems the ability to automatically learn and improve from experience. So, by solving this droplet problem, it will help future researchers in their application of artificial intelligence also. And we want to be able to train our computers to think for themselves, essentially. Research goals. Our long term research goals are being able to identify bad cell types that may have disease cells, such as cancer, also to use microfluidic devices to help identify cancer cells. Our short term goals are to use deep learning techniques like CNN. And what CNN is a neural network that acts like our brains to help process information such as image processing, image classification, and image recognition, which ties into deep learning because deep learning is when a network is capable of learning data on its own so that it will know, okay, this is an embryo or droplet, and be able to differentiate between the two. And we also want to be able to create a code to identify the number of droplets generated.
Moving on to the objectives of our research. In this research, we aim to learn how to train a neural network understanding deep learning, which can also be shown as a general idea in this figure below, and to apply the knowledge from the embryo analysis to the droplet generation. The deep learning shown in this flowchart shows us that a set of data, in this case, it is a set of frames with a car in blue in each frames, are used by the convolutional neural network, in which in deep learning, convolutional neural network is a class of deep neural networks most commonly applied to analyze visual imagery. So this network will analyze the car's features that were similar to each frame, such as the outline and the dark blue color of the car itself. And it will help the network understand and recognize these features of these cars. So once a new set of data was given to this specific network, the network will recognize if this is a car or something similar which is a truck or a bicycle, in which in this figure, the network fully understood the features of this set, and it was successfully, for 95%, successfully recognized the car. So, for our approach and design, in our research this summer, we only have to focus on three major steps. It's to create, train, and analyze. First, to create a data set in which involves extraction of frames from each droplet generation videos and labeling each frames by segmenting the droplet and background, which can be seen in this figure in the bottom left, where the droplets are selected as pixel labels in red and the background in blue using the image labeling app in MATLAB. Then, after creating a data set, we need to train our network and let it differentiate the droplets from its background. This might take a while depending on how much frames you are using as a data set, in which in our case, we only used 300 frames but still took us 10 hours of nonstop training. And finally, to analyze the droplet generation videos using the network trained through the droplet analysis prototype that we coded based off the embryo analysis software code that the previous researchers had made. This lets the code analyze or extract frames and segment the droplet on each frames from different droplet videos, which can be seen on the bottom right figure. Unfortunately, since we only used 300 frames for our dataset, some droplets were not recognized fully in each frame, such as this specific droplet, as you can see in the bottom right corner of this figure. And if you can visually see that there are some parts that were not recognized in this specific droplet. And so, as part of our research, we had to debug some parts of the existing codes as we tried to recreate this for the droplet generation analysis. An example for this issue is these figures that are comparing the original code to the revised versions, where we had to change the file path and the slashes for Linux, Mac, and other platform or devices users, in which the current researchers are using Mac. We had to use the forward slash as a separator for any file paths used in the code. Meanwhile, for specifically the Microsoft platform users, they had to use the backslash, which can be shown in line number 2 in both codes. Meanwhile, another set of revisions that we had to create or made, which can be seen in line number 5, we had to manually input the name of the file instead of calling the variable name in the function called video reader, which you can see in line number 5. Also, we had some trouble finding the appropriate codes to use for some specific task. Thus, we had to compile these files properly.
Regarding our future work, we want to be able to successfully tell whether cells inside this droplet are good or bad disease cells so that we will be able to help catch disease cells at an earlier stage, like mentioned before. So, in this picture, you can see we have a screenshot from a video we extracted, and these are droplets, the big circles. And inside those big circles, you see they look like air bubbles, and that is what inside those are cells inside this droplet. So we want to be able to determine if there's cells inside it, and if so, how many, and how many diseased or bad cells there are. Issues that we've had were because of COVID and due to the high data that we have to run through MATLAB using our computers aren't built for this type of training and material. So we've used TeamViewer to remotely control our lab's computer for better results. We also debug codes using MATLAB, and in the end, our solution We want to be able to count and identify cells inside a droplet successfully, which we should do because we've learned from research so far that by using MATLAB and its applications, it will help us generate reliable data to test our codes and debug codes if necessary.
And in conclusion, this research helped us recognize even further importance of embryo analysis and droplet generation. And how can we further improve our analysis code? Also, this helped us learn how to train neural network using deep learning, which helped us develop a functional code, which also a prototype code for the droplet generation analysis, which we hope that it could be further developed and analyzed further the droplet generation videos with the i- ability of counting the number of droplets generated, the number of cells in each droplet, and be able to differentiate the good cells from the bad cells.
One. And lastly, we just want to say mahalo to the Native Hawaiian Science and Engineering Mentorship Program, PEACE 2, MDC, Freshman Bridge, which are the programs we fall under, our supervisor, Dr. Aaron Ota, our mentor, Minakshi Vora, for taking us in and allowing us to intern with them this summer for this research project, the University of Hawaii at Manoa, the U.S. Department of Education and HEI Charitable Foundation, the National Science Foundation. Thank you all for being our sponsors and allowing us to have this opportunity.
And for everybody listening to our presentation, if you do have any questions, please leave them below and we will answer them as soon as possible. Mahalo.
Thank you.